next two mechanisms we need to describe are transcription and translation. In translation, we're going to interpret the working blueprint made in transcription. Transcription is the process of making the working blueprint in the form of RNA from the original DNA hereditary master copy that we find in every cell on Earth. When scientists were trying to determine how cells use DNA to make more pieces and parts of the cell, it wasn't too difficult to finally determine that the mechanism of creating the working blueprint, transcription, was essentially making an RNA copy of DNA. They also noted that in order for the flow of information to continue into a working model, or in other words, creation or synthesis of new pieces and parts of a cell, we had to interpret that working blueprint into the form of a protein. Now, this is called the central dogma of gene expression. DNA to RNA to protein. Now, can this process be reversed? For quite a few years, actually several decades, scientists did not think or could not find any mechanism that would allow us to essentially go backwards from protein to RNA or from RNA to DNA. But in the 1980s, a new enzyme was discovered from a virus called reverse transcriptase. The viruses were retroviruses, and the enzyme reverse transcriptase is a naturally occurring enzyme that the viruses use to make a DNA copy from an RNA original. We'll talk about reverse transcriptase in more detail when we talk about the retroviruses and when we talk a little bit more about genetic engineering. But the central dogma of genetics was thought to be the only way that information could flow from DNA to RNA to protein inside of a cell for quite a few decades. Transcription in a prokaryotic cell occurs in the cytoplasm of the cell. In fact, all of the mechanisms, transcription and translation, occur inside the cytoplasm of a prokaryotic cell. So both mechanisms occurring in the cytoplasm in prokaryotes makes things fairly simple and straightforward, and they made excellent models when scientists were first trying to learn how transcription and translation interrelated with one another. Transcription and translation in a eukaryotic cell are in distinctly different compartments. Transcription occurs inside the nucleus, where the RNA copy of the master DNA blueprint is made. Now, the first primary RNA transcript that's manufactured or synthesized in transcription in the eukaryotic nucleus isn't fully functional. In fact, there's a fair amount of what's called post-transcriptional modification. Post meaning after, transcriptional during the process of transcription. Modification means we have to change it, has to occur to the RNA transcript before we can actually use it in the next mechanism of translation. Now, the post-transcriptional modification occurs in the nucleus. We're going to have to essentially remove some non-translatable bits and pieces of RNA. In fact, originally when scientists tried to take an RNA transcript of eukaryotic DNA in a test tube and use that original primary RNA transcript to make some kind of a protein in translation, they essentially got garbage. an unusable protein. So they had to go back and more closely examine what was happening inside of the nuclei and inside of the cytoplasm of eukaryotic cells. Lo and behold, they found out that the primary RNA transcript of a eukaryotic cell typically has both usable, translatable bits of RNA called exons and other bits called introns that cannot be translated. The introns are actually parts of the area of DNA that are regulatory sites, and we have to remove those in order to create a fully functional piece of what eventually will be messenger RNA or mRNA. Now, the introns are removed, and we're going to have to add a 5' cap made of guanosine or guanine, and we're going to have to add a 3' polyadenosine tail. Once we have our 5' guanine cap and our 3' poly A tail, and we've removed the introns, now we have a fully functional piece of mature messenger RNA that can be translated in the cytoplasm, which is where translation occurs in a eukaryotic cell. So there's far more detail, far more sort of fine-tuning of transcription that has to occur in a eukaryotic cell before translation can occur, which makes prokaryotic cell models much easier to study, at least when we're starting to try and work out some of the fundamentals of the mechanisms of transcription and translation. Of course, if we want to know more about eukaryotic cell models, we have to go to the eukaryotic cells and patiently work out all of the fine-tuning, all the fine steps for transcription and translation. Now, the ribonucleotides, similar to the deoxyribonucleotides, are in the triphosphate form when we're using the free monomers to create our RNA copy. And as we did with the synthesis of DNA, we're going to use the potential free energy in those high-energy phosphate bonds to assist in the synthesis of the RNA copy of DNA during transcription. In the synthesis of RNA, we're going to use purines and pyrimidines along with the ribose and phosphate groups, just like we did with DNA, with one small exception. For the purines, we're still going to use adenine and guanine. For the pyrimidines, we're going to use cytosine, and instead of thymine, we're going to use uracil instead. So just like we did with DNA, we're going to pair guanine with cytosine. But for RNA, instead of having AT pairs, we're going to have AU pairs. Adenine is going to hydrogen bond to uracil when we're making RNA copies of DNA. Just like replication, we're going to have an initiation, elongation, and termination phase of transcription. We won't need to learn that much detail about the three different parts of transcription, only that we have a primary enzyme called RNA polymerase that's responsible for the elongation phase, or what we'd call the polymerization phase. RNA polymerase is responsible for pairing up ribonucleotides with the DNA template to make the RNA copy. Now, in transcription, there are various different types of RNAs that are synthesized. Messenger RNA is just one of several different types of RNA. In addition to mRNA, which is messenger RNA, we're going to find that in transcription, cells also make ribosomal RNA, transfer RNA, and even a little bit of RNA that has enzyme function that we call catalytic RNA, or sometimes called ribozyme. All of these different types of RNA are then going to be used in the next mechanism of translation. In order to interpret the working blueprint of messenger RNA, we're going to have to know what the encoded message is. That's what this genetic code can be used for. Notice that we have all 4 possible ribonucleotides and every possible combination of 3 ribonucleotides or triplets listed on the chart. Each triplet of ribonucleotides is called a codon, and each codon represents at least one amino acid, and in fact, most amino acids have multiple codons that can be used to request the placement of that amino acid at a particular position in the amino acid sequence, or primary structure of the protein. Now, the genetic code is almost universal, which means if we have messenger RNA from 100 different organisms, we should be able to use this genetic code to reasonably successfully predict the primary structure of any protein that's being created from those different kinds of messenger RNA. Now, we say almost universal because there are a few rare bacteria and archaea that have a few substitutions or different amino acids required for certain codons. Not all of the codons, just a few, which means even with those slight differences, we still should be able to roughly estimate what the amino acid sequence should be for virtually any kind of protein, as long as we have the messenger RNA from the organism. Now, in addition to being universal, we're also going to see that the genetic code has a few other characteristics. The genetic code has punctuation. There is one single codon that is used as a start codon, and that's AUG. Now, the codon AUG actually does require the placement of an amino acid called methionine wherever we find that codon. But if we have other features on the message that clearly show the mechanisms of translation, which is the next mechanism we'll talk about once we get through describing the genetic code. In translation, if we have the right information just prior to what's going to be recognized as a start codon, then methionine is put in place and the machinery of translation knows or interprets the material needed to start translation. This will make a little more sense when we see the illustration. But in other words, it's just like the first word of a sentence. The first letter has to be capitalized. Well, we really don't know that it's the first word in a sentence unless we look at the context. What other information or images are either before or after that first word of the sentence? The same thing's true of the start codon. There will be other nucleotide sequences just before we get to this start codon that tells the machinery of translation this is where we start. If we find the AUG somewhere within the message, and it's simply part of the whole string of amino acids that we're assembling for a primary structure of a protein, then we're simply going to incorporate the AUG methionine as a regular amino acid in the primary structure. In addition to a start codon, we also have 3 stops. Just like we have punctuation at the end of a sentence, These 3 stop codons do not require any particular amino acid. It simply is going to end or terminate translation. A couple of other features of the genetic code. The genetic code has wobble in position number 3. Let's take a look at the 4 codons that could be used to request the amino acid leucine in our messenger RNA sequence. Notice that the nucleotide that's used for positions 1 and 2 are exactly the same. The nucleotide in position number 3 could be any one of the 4 possible ribonucleotides used to create RNA. That's the wobble of the genetic code. It really doesn't matter what nucleotide we put in that 3rd position. As long as the 1st and the 2nd are the required nucleotides, we still get the same amino acid. The fact that we have multiple codons that could be used for the same amino acid is the last characteristic of the genetic code, and that's the degeneracy of the code. We say that the genetic code is degenerate because we have multiple codons that could still be used for the placement or the use of the same amino acid. And for leucine, we actually have 6 codons that could be used in a piece of message and still get the leucine amino acid that we would need in the construction of our protein. So the characteristics of the genetic code: universality, punctuation, wobble for position number 3, and degeneracy for multiple codons for the same amino acid. Okay, now we can assemble all of our components for translation. We're going to use ribosomal RNA that's found in the construction of a structure called a ribosome. We're going to use transfer RNA, like a truck or a transport device. And we're going to use messenger RNA, which is our working blueprint. This is an illustration of how complex a ribosome actually is. Using both protein and RNA, nature assembles all these components into a 3-dimensional structure, very much like a protein factory. In fact, ribosomes are often described as the site of protein synthesis. Here's another illustration of the typical ribosome. Ribosomes come in 2 parts, a large and a small subunit. It just so happens, if you recall, that prokaryotic ribosomes and eukaryotic ribosomes that we've studied previously when we looked at structures for protein synthesis, the small subunit is going to be about a 30S size, which means particular centrifugation size, and the large subunit is described as a 50S subunit. We have 3 seats, an E site, a P site, and an A site. Those are going to be locations for the placement of a transport device called a tRNA. Notice at the bottom, almost like a railroad track, we have a site for messenger RNA binding. The ribosome, almost like a locomotive chugging down a track, is going to move down the message 3 nucleotides or 1 codon at a time to finally reach the end of the track, and through that process synthesize a new protein. This is an illustration of another component that's going to be used in translation. It's the transfer RNA. Now, the two-dimensional illustration of transfer RNA, it makes it look like a three-leaf clover. We have the three loops and a stem. The most important parts for us will be the stem and the loop that's the farthest distance from the stem, labeled anticodon. Now the stem, where we have the 3' hydroxyl, is going to be the location for the attachment of a single amino acid. And that's part of the tRNA job. tRNA functions as a transport device. It's going to transport amino acids to the site of the ribosome and the messenger RNA and help line up those amino acids so that peptide bonds can be formed between each amino acid to start forming peptides or proteins. Peptides and proteins are the same kinds of materials. They're simply different names for the same thing. A peptide is made of amino acids. A protein is made of amino acids. And that's going to be our product from translation. We're going to make proteins in that process of translation using all of these components: the ribosome, the tRNA, and the message. Now, the other part of the tRNA that's important, in addition to the 3' hydroxyl stem, where the amino acid is going to be located, is the anticodon. As the name implies, this is actually a triplet of ribonucleotides that are going to be complementary to whatever the codon is on the messenger RNA. We're going to find that we have to line up the tRNA next to the message. And in order to stabilize that structure just long enough for the amino acids to get bonded together, we're going to have to temporarily link the codon and the anticodon. So, for example, if we have a codon on messenger RNA that's GCU, then the anticodon would be a triplet of ribonucleotides that are complementary to GCU, which would be CGA. Just make sure that you remember which nucleotides or nitrogen pairs will go together: G and C, A and U. And it's fairly simple to match up the proper complementary nucleotides. Now, there are specialized enzymes that are responsible for attaching the proper amino acid to the transfer RNAs. Those enzymes are called aminoacyl tRNA synthetases. We have to have specialized enzymes to make sure the proper amino acid gets attached to the correct transfer RNA, because remember, the tRNA is going to line up with the codon. So very much like the genetic code, where we have a specific codon that's going to be used for the placement of a specific amino acid, well, the anticodon has to properly match as well. And that's what the aminoacyl tRNA synthetase helps to do. Now, when we have the amino acid attached to the tRNA, we describe that tRNA as charged, meaning it has its amino acid package attached and it's ready to go to the site of protein synthesis or translation. When we remove the amino acid and the amino acid is no longer found on the tRNA, we describe that tRNA as uncharged. So a charged tRNA has its package, its amino acid, ready to go to the site of translation, also called protein synthesis, where it will drop off that package, and then the uncharged tRNA is going to recirculate, go back to an aminoacyl tRNA synthetase to get another package or another amino acid attached. This illustration has a transmission electron micrograph at the top, and then a cartoon drawing corresponding to what's illustrated in the real photo at the bottom. This is transcription and translation that occurs almost simultaneously in the cytoplasm of a prokaryotic cell. In the photo at the top, there's a very thin, almost closed line, sort of dotted line, sort of through the middle, going through the length of the picture. As the drawing at the bottom shows us, this very thin little closed line is actually DNA. And RNA polymerase is traveling down the DNA, transcribing it all the way along. In fact, multiple copies of RNA polymerase are doing this at the same time. Now, as RNA polymerase is transcribing the RNA copy of the DNA, this illustration is showing us that it's messenger RNA that's being transcribed. There are all other kinds of RNAs that can be transcribed, but this is what's happening here. And we know this is messenger RNA because of the dark, large globs that are attached to it, which are the ribosomes. And of course, if the ribosomes are attached to our little string of RNA hanging off of our DNA clothesline, then we must have not only transcription occurring, but translation occurring at the same time. Or virtually, as soon as the message is made, it's being translated. Notice we have polyribosomes, or multiple ribosomes, attached to the message, and vigorously translating the hidden working message of the messenger RNA into an amino acid sequence or protein. Just like replication and transcription, translation also has an initiation, an elongation, and a termination stage. For initiation, we have to form what's called an initiation complex to get the process started. In elongation, we're going to assemble amino acids together, linked by peptide bonds, to form the primary structure of a protein. And for termination, when we get to a stop codon, translation gets terminated, and all the components essentially fall apart and can then reassemble in another time to be reused again. Here we have, in the sequence of cartoon drawings, the formation of the initiation complex. We're going to need a small subunit of a ribosome, the messenger RNA, and the first tRNA, which has the proper complementary anticodon to the start codon. And we'll need to line all of these parts up. The tRNA with the proper anticodon is carrying the appropriate amino acid, which for a prokaryotic cell is not just plain old methionine, but 4-mil methionine. That's FORMYL. A 4-mil functional group is a unique modifier that we find on methionine amino acids in prokaryotic organisms. Now, we have to have assistance from additional kinds of proteins that I often describe as pilot proteins, and they're called initiation factors. Initiation factors help guide all of these components, the message, the small subunit, and the tRNA, guide them and line them up. In fact, this is quite an expensive, energetically requiring process. Lots of helpers, lots of additional proteins, lots of energy needed for this whole process of the formation of the initiation complex to occur in the proper sequence. Once we've lined up the tRNA and the anticodon and the codon match, notice that the codon is AUG. The anticodon would be the complementary ribonucleotides, so it would be UAC on the anticodon. And we've now allowed the large subunit to line up. Now we can get going with the elongation phase of translation. Now we can enter into the elongation, or what's often called the translocation process. Notice that we have the fully assembled ribosome. We still have the first tRNA sitting in the P site, and now a second tRNA that has the appropriate anticodon that's going to be complementary to the next codon on the message is lined up with the assistance of an elongation factor. In fact, there are several elongation factors that are going to help to line everything up properly. So in the next little frame, we have two charged tRNAs sitting side by side. The first one's in the P site, the second one's in the A site. We're going to have an enzyme that's found on the ribosome called peptidyl transferase that will form the peptide bond between the two amino acids. And so we now have one uncharged tRNA, and the other neighboring tRNA that's in the A site now has a dipeptide attached to it. And we have everything shifting down one spot. That shifting is called translocation. And we simply have shifted the seating position, or the seat, of the tRNAs so that now the uncharged tRNA is sitting in the E site, and the charged tRNA that carries two amino acids now instead of one is in the P site. Now at any one point in time, only two out of these three seats can be occupied. There's just not enough room for all three of them to be filled. So it's just like musical chairs. We're shuffling down the message one codon at a time, transferring the growing amino acid chain from one tRNA to the next as the peptide bonds are formed, going essentially from A to P to E, accepting a new tRNA, attaching those peptide bonds to the appropriate amino acids, and then removing those uncharged tRNAs at the exit site. Translocation and attaching amino acids one at a time to the growing polypeptide chain, or protein, continues until a stop codon is reached. When a stop codon is located, there is no tRNA that will sit in that site. Instead, there's what's called a release factor, an additional helper protein that will line up on the ribosome in that A site and help to disassemble or tear everything apart. So once we have the release factor occupying that last A site, the small and the subunit, small and large subunit separate, and the tRNA separates.